Hello and welcome to our session today. Today we are looking at pharmacodynamics. In this lecture, we'll be looking at the introduction to these concepts, the four common receptors involved in the process, then the intrinsic receptor interaction, the antagonist-agonist concepts, then drug potency, efficacy, and therapeutic index will also be reviewed in these sections. All right. So welcome to this session. Let's do it together. Previously, we talked of pharmacokinetics, of which we say that this one normally describes the actions of the body on the drug, whereby once the drug is ingested, it will be absorbed, it will be distributed, metabolized, and finally eliminated from the body. Now, in pharmacodynamics, we are going to look at what now the drug will do to the body, okay? And in the same process, we'll be looking at efficacy, potency, toxicity of the drug, okay? Remember, the drug, once taken, it has to get into the blood, and then from the blood, the drug will concentrate at the target site where we're going to have an effect, and this will be reflected in the body's clinical response. All right. So drugs normally enters the body and then it interacts with the bodies through the cell receptors. The interactions between the drug and the receptor normally lead to signal transduction. Okay, this signal transduction has some biological effect, of which some of them could be the therapeutic or the expected outcome for this type from this type of drugs. All right. So these signals are going to direct several actions in the body. Like they could even tell the DNA to stop replicating, okay, in the case of those drugs that direct things to be done in such way. Or they could result in several actions that are going to have therapeutic effects on the body along with side effects and other adverse effects. So generally, what we need to know is that drug normally interacts with our bodies through the use of the receptors. And we have different types of receptors, all right? But we have four major receptors that have therapeutic relevancy. And this include number one, we have the ligand-gated channels. Good examples of drugs that could use this type of receptors could be like the diazepam. We have the G protein coupled receptors, also called the 7-transmembrane protein. And then we have the enzyme-linked receptors, and lastly, intracellular receptors. So I beg we start with the first one so that we can be able to see what happens in this kind of interaction. Well, for the ligand-gated channel, these are just simply chemical signals. So in this kind of receptors, you find that it has a ligand binding site, right? So when a ligand binds to this channel is going to open briefly. All right, we have that site. So when the binding happens, this channel is going to open, and then therefore ions like sodium, potassium, chloride, calcium will be able to penetrate through this membrane into the cell cytoplasm. And then when you have now this once in the cytosol, they are able to bring about the cellular response. Once this is done, the gates usually close. Okay, and these are several examples. I will have several examples of drugs that normally use these concepts, okay, like the calcium channel blockers. All right, we can be able to see how they normally block the L-type channel so that sodium or calcium will not be able to get in, right? A link will be popping up. You can use it to do that, but at this juncture, we just want to understand that there is a ligand-gated channel ion in which this channel has a binding site. So this is where the drugs or the pigments are going to attach. Once they attach, they open these channels, and then these channels will allow the exchange of these electrolytes, which are going to cause this balance that is caused, either depolarization or repolarization for that case. Number two is the G protein coupled receptor. and this is also referred to as 7 transmembrane protein. And you can count it with me. We have 1, 2, 3, 4, 5, 6, 7. All right. So it is called so because it passes through the cell membrane 7 times. All right. These receptors, along with the enzyme-linked receptors that we shall be looking in at a short while, have the ability to amplify signals. All right. They do amplify the signals. So that means if you just have one stimulated G protein receptors, it can activate many adenyl synclases, which can result in more cyclic AMP molecules produced and ultimately amplified response. So this G protein coupled receptors is composed of 3 subunits, which consist of the alpha, the beta, and the gamma receptors. All these are the G protein. All right. So in its active form, you find that the alpha receptors, they have a GDP, right? They have a GDP here, guys. So when there is a ligand which comes and attaches to this receptor, the affinity now for GTP will increase. Okay, affinity for this GTP will increase, and this now leads to replacement of the GDP with the GTP, okay, for that case. So this in turn will cause the alpha subunit to dissociate from the beta gamma complex. Then both of these complex now will go and interact with other enzymes. So guys, you can be able to see here now it has dissociated. So we have alpha, and then now we have this other complex where we have beta gamma complex, of which the two now will go and now bring about the cellular responses in the cells. All right. Okay. So you find that G protein receptors are several, but the ones that you're supposed to remember are mostly 3. We have the GS, GI, and GQ. GS has a stimulative G protein, in that it will activate this enzyme we refer to as adenyl synclase, and this adenyl synclase will produce cyclic AMP from ATP. And as you all know, cyclic AMP is normally a second messenger. So the GS, all right, normally ends with a second messenger in the name of the cyclic AMP. Now, GI, on the other hand, it has an inhibitory trait, in that it will inhibit the adenyl synclase, and thus this will lower the levels of CAMP in the cell. Lastly, the GQ normally activates a class of enzymes that we refer to them as phospholipases, C, and we can abbreviate them as PLC for our discussion in this case. So all these 3 are major G protein receptors that you are supposed to remember, and you can see how they are spread all over the body. All right, and it is in this spread that they are now able to bring about their effects in cells. All right. Okay, guys, we can move to we can look at the phosphodiesterase C, or the PLC. These proteins, they normally produce 2 second messenger, one in the name of DAG and another one in IPL3. Right. So DAG, this is the diacylglycerol. Okay, it behaves the same way like the cyclic AMP, in that it leads to different responses through activation of the protein kinases for that case. All right, activation for protein kinases. And we have now the the inositol triphosphates, and this one, they produce various responses by mediating intracellular calcium response. You can see it's releasing calcium ions in this case, and in that case they are able to lead to neurotransmitter release in cell proliferation for that case, along with the DAG. Okay, so those are the 2 second messenger that can be derived from the phospholipases C for that case. Okay. The third group of receptor is enzyme-linked receptors. So this one, just like the G proteins, they also have an extracellular binding site, right? Binding site where a ligand can easily attach then stimulate enzyme activity inside for that cell, right? You can see, so these signaling molecules could either be a hormone or growth factor. It will attach here, then it can bring its effect for that case. A good example for drug that we normally use this concept is the insulin hormone, right? The insulin hormone for that case. So it's also good to know that most enzyme-linked receptors are made up of tyrosine, all right? And this means that they display kinase activity, and there is an amino acid tyrosine that is always involved for that matter. So when a ligand binds to this, to the two of these receptors, it will cause conformational changes that will result into aggregation of these both receptors for that case, and that brings about the therapeutic differences. So once this dimer is formed, then tyrosine regions will get activated and cause and now bring and cause the ATP to become ADP, okay, and which is going out result into auto phosphorylation of receptors. Remember, once each tyrosine picks up a phosphate groups, different inactive intracellular proteins will come up and attach themselves to this phosphorylated tyrosine. So this move causes changes in the attached protein, which ultimately leads to cascade of activation that normally produces a cellular response in different tissues. So we could have some differences between the two, where we have G-coupled receptors, enzyme-linked, and the ligand channels. So one of the major characteristics or similarity between the three is that all of them, they have extracellular ligand binding region, right? For that case. For the transmembrane region, the G, we have said there are 7 for that case. And phosphorylation is required, but it's not intrinsic for that case. Then we have an alpha subunit of G protein. In terms of onset of action, you find that out of the three, ligand-gated channels are the fastest, okay? We'll have a quicker response in these ligand-gated ion channels, followed by the G protein coupled, and lastly the enzyme-linked receptors, okay? The last one is intracellular receptor, and the reason why this is separate is because it is somehow different from the ligand channel, from the G protein, from the enzyme, in that this one, they will be entirely found inside a cell, all right? You can see they are found inside the cell and not outside as the other three. So this means that for the ligand to get to the receptor, it must first of all cross the cell membrane, and this cell membrane, we all know it's lipid in nature for that case. So then once it's inside, it can now bind to this receptor now, okay? Then activated receptor complex can now move into the nucleus, so it will now move into the nucleus and bind to the DNA and regulate gene expression. So this will ultimately lead to synthesis of specific proteins, all right? So that is for the intracellular receptor. So those are the 4 different types of receptors that are very key in pharmacodynamics. So let's look at also now about the life cycle of receptors. So you find that each cell DNA contains a code that will be used to synthesize proteins from different receptors assembled for action. So once they have been assembled, the receptor will get embedded into the cell membrane and can now receive and respond signaling molecules, which in most cases could be hormones or enzymes for that case. So if the cells receive too much stimulation, you see they could damage. So just to protect itself, the cells can down-regulate receptors, meaning they can take them out of the membrane and recycle them, which leads to fewer number of expressed receptors. And this could lead to decreased sensitivity to signaling molecules for that case. If most of the cell receptors get blocked and the cell is receiving very weak signals, then the cell also has the ability to upregulate their receptors. So this means that more receptors can be inserted into the membrane, thus increasing cell sensitivity to signaling molecules. you have to know that generally that as the concentration of a drug normally increase, its pharmacological effect will also increase until we reach to a point where most of the receptors have been occupied for that case. So if you try to plot a graph where you are looking at the drug concentration vis-a-vis the pharmacological effect, you'll find that as we increase the dose, also the max effect also increases, okay, until we reach a point where all the receptors are assumed to have been occupied for that case, all right, in that manner. So when you try to get a half of this, okay, where we get a half of the response of pharmacological response. Remember, this is 100%, so all the receptors have been occupied. So this is the maximum effect of the drug, okay. Now we want to take it a half of it, so will be 50%. And if you try to draw it, we end up at the EC50, all right, the EC50. So this is the concentration of a drug that produces 50% of the maximum effect. The maximum effect is 100, so the half of it is 50. So the concentration of the drug that produces 50% or a half of the maximum effect, this is what we are referring to as the EC50 of a drug, okay? The concentration of a drug that generates 50% of maximal effect. This point tells us how potent the drug is, okay? If you want to know how potent the drug is, we look at this EC50. Remember, as you try to reduce the EC50, we say that the drug is more potent, all right? More potent for that case. So the more potent the drug is, the lower EC50 is, right? That's what we really need to know for that case. So that means it's required to, this EC50 is the one that is required to produce 50% of the pharmacological response, okay? If the pharmacological response is 100, the EC50 will be used to produce this 50% of what? Which is this for this drug A, which is here for this drug B, which is here for the drug C, and which is here for the drug D, okay? Okay. So as you continue decreasing, if a drug you are going to decrease EC50 and you still get the 50, that means that drug is more potent for that case, all right? Okay, so in this case we could see that A is more potent than B compared to C and D, right? So in short, like drug D, we need more dosage, especially let's take between 10 to 100. We need this quantity of drug to produce 50%, okay, as opposed to drug A, where we need less than 1 to produce the same effect, the 50%. So drug A is more potent than drug D because we need lower dose of EC50 to produce the same 50% effect by drug A. All right? Okay. That is about potency of the drug. We look at the EC50 of a drug. What about the efficacy of the drug? Look, now we can try to look at my diagram here. You can see we have drug A here, okay, where it has reached the maximum effect to be here. And then as you continue climbing the y-axis, you find that drug B has gotten to this level here. Okay, if you compare the drug B and drug A, which one is at a higher level on this graph? Drug B is at a higher level. You can be able to see here at the curve, and this one is at this region, all right? So that's why we are saying that drug B is more efficacious than drug A because this drug B reaches a higher pharmacological effect compared to the drug A. And we are saying higher just based on the Emax of the drug, okay? This one is at a higher level compared to the A, right? So that's why we say now drug B is more efficacious than drug A for that case, okay? Okay, so next we look at now the intrinsic activity of drugs, that the concept of agonist versus antagonist. Of course, we know the intrinsic activity, that is the just ability of a drug to produce its maximal effect. So we know if a drug binds to a receptor and then is able to produce maximal effect that is somehow comparable to the effect produced by our own body's endogenous ligands or substrates, and that means this will be an agonist, right? So an agonist will be able to bind, then bring about the effect of the drug, okay? So example of a full agonist could be like the Lasix, okay, the furosemide for that case, okay? But so what happens if there is no agonist, no drug, okay? We say that's now the basal effect, okay? Where there is no agonist, we only have the natural pigments available. That will be the basal activity. But immediately you get in another agent, and that's now will be an agonist, okay? So in the presence of a full agonist like furosemide, we could see maximal effect for this one, okay? So you can be able to appreciate the full agonist, and then now we have this one that will cause minimal effect, that is the partial agonist, before we look at the other ones, all right? Okay, guys. So partial agonist, we say that it will bind to a receptor at its active site, but will only produce partial response, all right? Partial response. Can you imagine? This is the endogenous agonist. That is how it will do. But if you give a drug that is a partial agonist here, now this is what the effect you'll see, okay? It's not as much as this other agonist, right? So partial agonists, they are unable to produce maximal effect even if they occupy most receptors. Okay, we have an example of a drug which is antipsychotic, that is ariprazole, okay, which is a partial agonist for that case. Next, we have inverse agonists. These are agents that will bind to the receptor, but instead of activating them, it will stabilize this receptor in their inactive form. So they are called so because they eliminate the basal activity, right? The basal activity is eliminated for that case, okay? It binds to the receptor, then it inhibits that intrinsic activity. This is what we refer to as the inverse agonist, okay? Inverse agonist for that case. So and then inverse agonist is here, right? Inverse agonist is here for our case in this presentation, okay? Okay, so let's look at now antagonists, right? Antagonists. So antagonists, we could have competitive or non-competitive, okay? In competitive is like for this case here, where we are having a substrate binding is being blocked, okay? So the agonist and the antagonist, they do share the same binding site. For this case, you can see that's the same binding site. But for non-competitive inhibitor, you find that the agonist and the antagonist, they have different sites, right? Like you can see here is the agonist site, but now the antagonist will be here, right? The agonist will be here. So competitive antagonists bind to the same site as the agonist. This is what we have been able to look at here, but does not activate it, thus blocks the agonist action, okay? However, if you want to displace this competitive inhibitor, we could increase the concentration of the agonist, then it will be displaced for that case. But for non-competitive, it can't be displaced, okay? It can't be displaced because some antagonists, you find they can form covalent bonds with the active site on the receptor, and thus the process is blocked entirely. We have allosteric antagonists, where these ones are going to they are different from the rest of antagonists in that they bind at different sites from the antagonist, okay? Like here you can see the agonist will be binding here, but now this allosteric antagonist, it has a different site, okay? It has a different site. So just like with the irreversible antagonist, you find that the allosteric ones will also cause reduction in the Emax but no change in the potency for that case. The last concept is about the therapeutic index, and this one we know that people normally respond to drugs differently, like the dose that will be required to produce an effect in person X may not be the same in person Y. So the therapeutic index is important in that it helps us measure the relative safety of a drug for a particular treatment. It's simply the ratio of the dose of a drug that produces toxicity in 50% of population to the dose of a drug that produces effective response in 50%. If you want to get it, you simply divide by taking the ED, which we have said is the potency, divided by what? ED, okay, we take the one that is supposed to produce the side effects divided by the ED, which is the potency. Okay, so that with TD over ED, like in this case, the side effects will start being produced at 6. And the effective that's able to produce 50% response, it is at 2. So we take 6 divided by 2, you get 3, right? You get 3 for that case. So this is now the therapeutic range for this drug, okay? These are the safe zones, okay, where this drug can be used and is able to bring about the desired effect without the necessarily side effects. So guys, in general, we have different drugs that have generally narrow therapeutic index, like that we as nurses, we need to be monitoring during the care process. So these drugs could be like warfarin, which is an anticoagulant, digoxin, which is used in heart failure and atrial fibrillation, lithium used in treatment of bipolar disorder, theophylline and minophyllin used in treatment of asthma and chronic obstructive pulmonary diseases, phenytoin, the one that is used in treatment of epilepsy, tacrolimus, the one that is used in immunosuppressions and organ transplantation, carbamazepine, the CBZ, of course used also in epilepsy and bipolar, amiodarone used in ventricular arrhythmias and atrial fibrillation, and lastly, gentamicin, which is an antibiotic that is mainly used to treat the mainly gram-negative bacteria. Okay, guys, that wraps the end of the session for this presentation on pharmacodynamics. Remember to go to the description section and attend the quiz on the same. Thank you so much and see you.